Hello, everyone, and welcome to today's webinar, Feasibility Study on an Automated Targeted Metagenomics NGS Application for Rapid Detection of Bacteria and Fungi Directly from Specimens, presented by our speaker, Dr. Jennifer Dien Bard. Dr. Dien Bard is a professor of pathology with clinical scholar designation in the Department of Pathology, Keck School of Medicine, University of Southern California. She is also the interim chief of laboratory medicine and director of the clinical microbiology and virology laboratories in the Department of Pathology and Laboratory Medicine at Children's Hospital, Los Angeles. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com/AmpliSeq. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you may utilize this Q&A box as well, and we will assist you directly. Now, before we get to our speaker, I would now like to welcome Kevin Bryant from Thermo Fisher Scientific to give an overview of the custom AmpliSeq solutions they offer and to introduce our speaker. Take it away, Kevin.
All right, thank you very much. My name is Kevin. I'm on the global marketing team at Thermo Fisher Scientific. I manage the custom AmpliSeq family of products, as well as our next-gen sequencing platforms, including the Ion Torrent Genexus system. You'll be hearing more about these products and this NGS platform later on in the presentation, but I just wanted to give a brief introduction now to discuss our custom next-gen sequencing capabilities and services that we provide. First, just start with a question. If you're running a lab, what do you do when the NGS test you need doesn't yet exist? And this happens often for many reasons. I guess the answer simply is you develop a custom assay. So you need a flexible solution for targeted NGS that will address your highly specific clinical research needs. Some of the top reasons individuals need a custom NGS testing panel are here. So one, you'll need the flexibility to add new biomarkers to your NGS test as they're discovered, right? It seems like almost every day there's a new publication that comes out identifying a new biomarker. If you're using a fixed assay, it's difficult to adapt to those changes and advancements as they come. Also, you might be studying novel or emerging pathogens, or for example, pathogens that can vary, like in different regions of the world or in different parts of the year. All of these would require a custom NGS testing solution as well. Third, you might have region-specific needs for the targets in your panel. For example, some variants are more important to test, or some biomarkers are more important to test in one region versus another. So you might need to optimize or have a panel designed for the region that you are doing your testing in. And fourth, you might have multiple goals that you want to achieve in a single NGS test. And we'll be talking through some of these in the main part of the presentation later. For example, if you want to test all the bacterial and fungal species in a specimen, you might need a custom NGS test to achieve that. All right, now speaking about our custom Ion AmpliSeq solutions, we support multiple different application areas. There's many different clinical research applications that could benefit from NGS testing. I listed some below here. So really, to take the NGS testing in the direction you need a flexible solution that can go where you need to go. Some of the more relevant application areas that we support are clinical oncology research, transcriptome and gene expression analysis, inherited disease research, reproductive health research, and microbiology and infectious disease research, which will be the main part of the topic for today. it's one of our more interesting application areas. And to take NGS testing further and to get up and running as fast as possible, an additional advantage is to pair a custom AmpliSeq panel with other services offered by Thermo Fisher Scientific, including analytical validation services and bioinformatics services. Pairing these all together will give even better results. All right, and finally, before we get into the main part of the session today that you joined for, I want to just walk through a couple of the main features and benefits of our Ion AmpliSeq technology that really empowers targeted sequencing. So shown on the left here, we offer a scalable targeted NGS panel approach, meaning you can look at just a few targets or genes in your panel, up to hundreds or even thousands, right? So you can scale it up or down as you need. This Ion AmpliSeq technology is compatible with a very low amount of starting material, so as low as 1 nanogram of DNA or RNA, and assays can be quite high sensitivity. Additionally, you can design one NGS test that can look at single nucleotide variants or SNVs, insertions or deletions or indels, copy number variations, CNVs, or gene fusions, all in one targeted NGS test. In addition to those DNA variants, with AmpliSeq technology, you can also do transcriptome and gene expression analyses with the same technology. And combined with our NGS platforms, you can natively integrate AmpliSeq technology into our Gene Studio or Genexus platform for fast, easy, automated NGS testing, which can deliver NGS test results in as little as 24 hours. And that's all the way from a specimen through the report. So it can be quite fast and easy to use. Finally, when working with a company like us at Thermo Fisher Scientific, we have a very strong track record. We've designed, literally tens of thousands of custom targeted NGS panels, making us a very trusted partner. Like, we'll be here to grow with you for years to come. Additionally, we have a very strong global service and support network. So regardless of where in the world your lab is located, we have a team of field support staff to help you with your studies, answer your questions, and make sure your tests are stay up and running. All right, with that, I'm happy to pass now to Dr. Jennifer Dien Bard, who will talk about using a custom designed AmpliSeq panel that she developed for use on the Genexus system for the application of rapid microbial identification and detection.
Thank you for that introduction, and thank you all for joining us today for this talk. Here are my disclosures. I have nothing to disclose here. And so the objectives for this talk will be to understand the importance of rapid next-generation sequencing-based approaches to detect and identify microbes in the laboratory. To also learn how custom Ion AmpliSeq targeted NGS panels can be used alongside with the Ion Torrent Genexus integrated sequencer to deliver NGS tests within as little as 24 hours. And then lastly, to understand the performance of the new rapid microbial identification method and how it has the potential to be incorporated into standard microbiology testing. I'm going to start the portion of my talk just focusing on the clinical need for this application. Microbiology has evolved significantly over the past couple of decades in particular, and the infectious disease approach of culture has progressed and has been supplemented with molecular testing, either directly from isolates or from specimens. And in the recent years, this has progressed further into sequencing options, including Sanger sequencing, but also more interestingly, at least in my opinion, the next generation sequencing. So ultimately, as a laboratorian, we're always looking for that unicorn when it comes to diagnostic tests. We want it to be good, we want it to be fast, and we want it to be cheap. However, this can be quite difficult to really obtain in the diagnostic setting. And so typically you would, if anything, accept 2 out of 3, which isn't so bad. Currently, culture is cheap and good, but not necessarily very fast. Antigen testing is cheap and fast, but not necessarily the greatest when it comes to performance. And PCR may be considered are considered good, somewhat fast depending on the methodology. And so the question really is, where does NGS fit into this box here, or these circles here? It's certainly good, but how do we address the other factors into making it fast, in particular, which is an area that I want to focus on. So in 2021, I was fortunate enough to be involved in the development of this consensus study with the National Academy of Science. And our focus was to combat antimicrobial resistance and the protection of the modern medicine. Um, in the document, we included this figure, which really identified factors that contributed to antimicrobial resistance. And not surprisingly, one of it is just the overall misuse and overuse of antimicrobials. And the second part that I find also very relevant is the fact that there really is suboptimal rapid diagnostics available right now, and this does contribute and has been found to contribute to the overall increase in antimicrobial resistance. And the pandemic certainly contributed significantly to this as well. And in our report, we commented on how there were considerable presumptive antimicrobial use during the pandemic in patients that were diagnosed with COVID-19. And this was a very much of a just in case approach in prescribing. There was one study that was published in 2020 that reported that despite the fact that only 7% of COVID-19 patients had secondary bacterial infections, over 70% of which were treated with antibiotics. And then another study in India found that there was an excess of over 200 million doses of antimicrobials that were used during the pandemic. The report that we compiled worked off of the previous 5-year action plan and built on it and I wanted to focus on the 2 points that's in the pink box right there. One is to support innovation in developing and deploying diagnostic tools that would improve the overall diagnosis and treatment of the patients. And then lastly, to also be able to collect and compile data to have us better understand what diagnostic tools would be an appropriate option to meet this need. So there certainly has been an increased interest and even a little bit of FOMO when it comes to the utility of next generation sequencing for infectious diseases. And even though it's this bright and shiny new tool, it really does have to have the same approach when determining whether or not to use it for clinical diagnosis. And the benchmarks that applies to this is whether the result that would be reported would alter a patient's management and affect outcomes. Also, the emphasis on turnaround time is key to allow for timely patient management, because if you're on empiric therapy for a prolonged period of time and the results come out even longer than that, there's very minimal impact that can happen. And then lastly, to be able to be used as a tool that it can potentially avert any other testing that may be deemed unnecessary. And this, if anything, can help contribute to offset some of the costs of this molecular test. So because of this interest and my curiosity, in the past, about 3 or so years ago, I did send out a survey through the clinical microbiology listservs. This is through the American Society for Microbiology. And the question that I asked the laboratory directors was, does your clinical lab offer infectious disease NGS as a clinical test? And of the 104 respondents, there were only a total of 19 sites that reported out as yes, they did perform this in-house. And when you separated out the clinical labs, there was only 16% of those labs that offered it in-house. And not surprisingly for public health, there was 50%. So despite the fact that with the bulk of the labs saying no, my next question to them was, is this being sent out to reference labs? And the vast majority, more than 80% of which determined that, answered that it was being sent out. So some key takeaways from this survey was that there was only a small percentage of clinical labs that are offering NGS testing in-house right now for infectious disease testing. But however, there definitely is an interest and definitely a need, as more than 80% of these labs are sending it out to reference lab for testing. And if anything, this offers us opportunities to improve and to grow and to really answer the questions as how we could potentially bring this back in-house. The next generation sequencing has a number of different applications. One is whole genome sequencing for species identification directly from specimen or from isolates, which allows you to also determine resistance mechanisms, outbreak investigations, etc. The next mechanism, the next application is targeted next generation sequencing, mainly for the identification to the genus and species level of a variety of different organisms, depending on the target that you have included. And then lastly is metagenomics NGS, which is the unbiased approach, shotgun approach of detection of pathogens, where you can also detect parasites, etc. And so for the purpose of this talk, I will be focusing on targeted NGS because that was the approach that we pursued for our laboratory. And so one of the primary reasons why we looked into this was that there certainly, as I mentioned, there certainly was a need, and we were seeing this even in our laboratory where there was an increase in volume over time. And if you look at even in recent years, the increase was quite dramatic. And then, of course, it comes with an associated cost that we in the department would be having to take on. And so you can see even from FY22 or 22 to 23, is just a huge expansion in the interest and the orders of these tests. Looking back at some studies and the overall impact that these types of targeted 16S or other targeted assays had on overall patient management. There was one paper that was published in 2020 that's listed here that looked at, I believe, 607 clinical isolates, and where they offered culture, bacterial 16S, ribosomal RNA, PCR and sequencing, and then targeted PCR on these samples. One thing I wanted to just note about what they found in the study was that the specimen choice that offered the highest yield was pus samples, abscesses, etc., whereas the lowest yield was found to be CSF. And another study corroborated these findings where they also reported lowest yield to be CSF and blood samples. The interesting thing about the study was that it found that 16S ribosomal RNA PCR and sequencing detected additional organisms at set 47 compared to what was identified by culture and PCR. And so some of the takeaways from that paper that I summarized here includes the relatively low overall positivity rate of 12.9% that were positive. And I'll touch on that a little bit more later with some in the coming slides. But what also was reported was that 53.2% of the positives that occurred were new diagnoses that were solely made by the 16S ribosomal RNA. And of the 91 patients that had prescribing information, there was a change of antimicrobial change that occurred in 14.1% of these patients. So relatively low. And in the cases that were culture negative and 16S rRNA positive, the mean cost that they guesstimated or calculated that a 16S positive culture negative result equated to about 4,000 pounds for one prescription change. So because of the low yield of positivity and the cost of the test, that was the calculation, was that is about $4,000 a pop for any positive result that they identified. And one thing I wanted to highlight from that study was that the mean turnaround time was 13 days, which is significantly longer than what they would have preferred and what we would expect. And a quote that I included from the authors states that laboratories with core sequencing facilities typically have turnaround time of 16S PCR and targeted PCR of 24 to 48 hours, which reflects the potential impact of repatriation. So basically, being able to bring these types of tests, these molecular tests in-house, may allow them to shorten the turnaround time. And there was another study that looked at a retrospective observation of 310 samples in 268 patients, looking at targeted 16S and 18S rRNA testing. And overall, the positivity for just the molecular test itself was low. But same thing, the impact was also very minimal. There was only a change in antimicrobials in 6 patients, so 2.2%. And for these targeted fungal tests, there were no changes in antifungal therapy. They reported a turnaround time of about 7 days, which is still faster than the previous study, but they did also mention that they felt that the minimal impact on patients was attributed to the fact that the turnaround time was delayed. We did a similar study about 6 years ago, summarizing 78 samples that were sent for 45 patients to a reference lab for targeted 16S and 18S or ITS sequencing, PCR and sequencing. And we found very similar findings, 14% positivity rate, which is quite low. We found that there were a useful diagnostic input in 7 of these cases. However, there were only antimicrobial change that was made in 1 patient. So overall, low yield as well. Some considerations from our study, overall, there was a low impact on patient management, and we attribute it also to the fact that the turnaround time was 11.5 days, and even up to 14 days in some cases. And we commented on the lower positivity rate of 14%. And, some postulations was that, is it because of overtesting? Are we including specimens or cases where it likely is a non-infectious etiology? The probability of a test actually picking up a pathogen is quite low, which then dilutes, the true positive cases. And also, at the time, we're sending remnant samples in many cases that perhaps would have sat around in the refrigerator for a bit before it was sent out. So that could potentially have also contributed. So overall, there are factors that affect clinical utility of NGS. Case selection issue, the right population, as I mentioned, whether you would be sending it out after conventional microbiology results is available, treatment therapy and treatment failure potentially being a factor as to why you may send. Sample collection issues as well, the timing of the collection, the sample type. For example, sending out a swab for NGS is likely not optimal. Result interpretation as well. This is important because it is a more sensitive test. You may be picking up contaminants, dead microbes, etc., that may be considered a clinical false positive, but also clinical false negative in some cases. And then lastly, of course, the issue of expenses. It's not covered and reimbursed by insurance currently. And then, as I mentioned in some of the other slides, just the importance of the turnaround time. And I do think that there are improvements and opportunities for improvements of all of these factors. If a test can be brought in-house, it wouldn't necessarily address all the issues, but at least there may be more oversight with implementation of diagnostic stewardship efforts, the utilization of the medical microbiologist to help with the interpretation using in the context of the clinical presentation, which may not necessarily be available to the reference laboratory that are offering these tests. So what would be optimal? So as a lab director, what would I want to see when it comes to NGS and how I would be able to bring it into my lab? So optimally, I want it to be as close as possible to the clinical laboratory. And at this, using that term, I'm calling it the point of care. Um, but this is, of course, easier said than done. There are multiple hurdles, as as would be expected for any lab test, but more so for a highly complex molecular assay. So what are the problems that would need to be tackled for us to be able to even consider bringing this on into the clinical laboratory? And there are many problems, and I've only listed a few here. I'm sure there are many more, but ultimately the infrastructure. Does the clinical lab even have an infrastructure for pre-analytical, analytical, molecular setup? Molecular expertise, of course, is going to be extremely important. These are highly complex procedures that are involved, and it also requires extensive hands-on time to set up the library preps, etc. Clinical microbiology tends to not have a lot of expertise when it comes to bioinformatics, and so having access to that to help with the post-analytical process and to simplifying it so that it can be reported out in as streamlined a process as possible. And then, of course, as I mentioned before, the and the reimbursement is definitely an issue. And this slide just really illustrates the complexity of the different steps that are involved for any NGS application, whether it be whole genome sequencing, targeted sequencing, or metagenomic. And you may incorporate some automation for pipetting, but ultimately it still is a very manual process. And in order for it to be realistic or feasible to be brought into a clinical microbiology lab, a lot of these steps would have to be eliminated. So where we need to go, we need to go from the specimen, pop into the machine with minimal hands-on time and preparation of the specimen. Plug and play is really the key. We want it to be at a state where you can have small batches of samples that are tested, which will also improve the turnaround time. You don't want to necessarily, depending on the lab, but certainly from my lab, you don't want to necessarily have to wait for 96 samples to come in before you pop it into the machine either. So being able to have like those smaller sample volumes would be very helpful. And then for same thing from the informatics side, having a plug and play approach that would spit out the results where we can get the identification, depending on the approach, the potentially resistance profile, etc. That would be extremely helpful. And tying it back to stewardship, which is a huge passion of mine, really ultimately being able to bring it in-house, allow us to have like that direct oversight where we allow for the right test for the right patient and at the right time. And this is, the question for diagnostic stewardship in many cases is whether it's to test or not to test. And the microbiology laboratory play a key role in both diagnostic stewardship, but also in antimicrobial stewardship, which really all ties together. And I would argue that from the diagnostic stewardship point, having that microbiology expert, that medical microbiologist be that key person or help lead that committee to guide diagnostic testing selections would be extremely helpful for the institution. So you really want to find that sweet spot. You don't necessarily want everybody to get that test where it's a free for all, because of course there are considerations, including a negative patient impact. But you also don't want to be that grim reaper that does not allow any testing at all. So, wanting to kind of understand what that perfect sweet spot would be. So with that, when you're taking into consider what that spot would be, you want to maximize impact. And with that, it really is to have judicial use for only patients that have high suspicion of infection. And you can define this in multiple ways, perhaps in specimens with higher yield, such as organisms seen on a gram stain, but it's negative by culture. And then, of course, emphasizing that collaboration, because that is extremely important, and it doesn't help anybody to be siloed into your own sections only. And so the NGS playbook, as I put it into 4 rules, would be, to have an approval process for this, whether it be offered in-house or outside as a reference lab, restrict or testing on multiple specimens, and to reserve for difficult cases where perhaps standard of care testing have been negative, or critical cases where it will directly quickly have an impact on that patient. And then to emphasize that this is certainly not a rule out test in any way, and you really do have to correlate it with clinical findings and other ancillary testing. And tying in the results that will be coming out, it's just emphasizing that these are complex tests that certainly do need some aid in interpretation, and to utilize your laboratory experts, medical microbiologists that can really help with this. And so myself and Dr. Christina Castellas, who's a colleague of mine here at CHLA, worked on this review article where we, talked about potentially modeling it after oncology with their tumor board discussions. Perhaps we call it an abscess board, nodule board, pus board, whatever it is. But, it's to have like that direct communication with physician, avoid having our ID colleagues who we work really well with, but having to have them be that middleman so that you can directly, explain your reasonings behind certain things, and also the nuances of certain diagnostic tests and the interpretation of it. So collaborating in that way, discussing the sequence results, determining perhaps whether modifications of cutoffs can be had depending on the case, like all these things that could be very helpful in a case-by-case basis. And now we're going to shift to the feasibility of a new targeted metagenomic NGS test for rapid microbial detection and identification. I'd like to present the Ion Torrent Genexus system, which is an image of it here. This system offers an end-to-end workflow from the biological sample to the report. It is quite fast as 24-hour turnaround time, which allows you to provide those results. It is automated in the sense that there's a sample extraction, quantitation, library prep, sequencing, and analysis process. It also allows for the reduction in potential manual errors that would be had if it was a person that's going to have to do all of the pipetting, etc. And it also has the flexibility of economically running as few as one sample, reducing the need for batching, which is something that I mentioned earlier is a need, particularly for labs that are much smaller in size. The system itself and the consumables are manufactured at facilities registered with the FDA and ISO. It's 13485 certified. And the research applications, although, it's not as interesting as infectious diseases, just kidding, but there are other applications such as oncology, reproductive health, and inherited diseases. So we had the pleasure of using the Genexus system during the pandemic, where we were performing whole genome sequencing for SARS-CoV-2 for positive samples. And one of the reasons why the instrument was of interest to me was the automation capability, the minimal wet lab hands-on time, as depicted by this table that we published in JCM in 2022. It's 10 to 20 minutes of wet lab time versus we calculated about 245 minutes with the Illumina that we were also using at the time. And the turnaround time, because you can run smaller samples and because it is so automated, we were able to complete one sample within 44 hours from the time the specimen was received in the lab, and which would include having to perform the PCR to confirm that it is positive for SARS-CoV-2. So it was a significant game changer for us. So the next steps, after the pandemic, the approach that we wanted to look into was targeted NGS for bacterial, fungal, microbacterial. We wanted same thing to prioritize that automation that the GenNexus is capable of, which would allow for hopefully reduced costs when you think of labor costs, and then reduced turnaround time. So we named this whole study Amplified for automated targeted metagenomic next generation sequencing pipeline for rapid infectious disease diagnosis. And the entire study was performed on the Ion Torrent GenNexus system. And collaborating with the Thermo Fisher team, we worked on designing a panel that targeted both bacteria, including microbacteria and fungus. And this included 16S gene, but also species-specific gene that was developed by the Thermo Fisher scientific team, and then the ROBI for microbacteria. We also wanted to include fungi using the ITS and D1, D2 region. And our focus was to ultimately include specimens that we are currently sending out. So that included sterile body fluids, tissue, and then we wanted to also include isolates. Okay, so the workflow that we established is a automated DNA extraction system. And following that, and that takes about 15 minutes of hands-on time. Following that, we then had about 30 minutes of hands-on time to prepare the specimens and to load the reagents onto the GenNexus instrument itself. Once that happens, the rest of it is all internal, where there are automated amplification, library prep, and then sequencing all within the machine. So by this point in time, our staff could walk away. And then after that, there is the FASTQ demultiplexing adapter, so that occurs within the GenNexus itself. We then incorporated a CLC genomic system using from QIAGEN for the read filtering and clustering. And then we also, working with our informatics group, developed some tools to help streamline just the interpretation and analysis of the data itself. And so basically, what our very talented bioinformatician developed was this dashboard. And this is a snapshot which illustrates a very user-friendly interface for filtering and also prioritizing species identification that would deem to be true positives. And the species that were listed are on the left-hand side of the column. And the dots that are present here are basically indicated by size and color. And so the relative size of the dots correlate with the relative abundance of organism that was present, and then the color correlates with the specific reference database that was used to make this interpretation. And then for the study, within the study itself, we included 50 samples that were tested on the AmpliSeq panel. And looking at the retrospective data of these patients, we classified them as either not infected or infected. So there were 16 cases that we deemed to be in the absence of infection, due to the fact that they did not have an ICD-10 code that was infection related, other inflammatory markers, fever, etc. And then we had 34 patients that we deemed to be true infection, same thing based on some of those parameters. And this is a breakdown of the specimen type, with the majority of which being sterile fluids, followed by tissue. And then breaking it down further, we compared it against conventional culture. And I'll have another slide that will summarize this, but for this particular slide, I wanted to focus on the last portion, which is the concordance between infection status and the metagenomic NGS test that we used. There were a total of 17 that were culture positive and metagenomic NGS negative, and then there were a total of 30 that were culture positive and also NGS positive. Focusing on the ones that were that were discordant. The 7 that were culture positive but NGS negative were actually found to be common contaminants and insignificant. So that was great. So if anything, NGS correlated more with clinical findings than the culture did. For the 10 that were true infections, but NGS missed, the majority of them were very weak positive with like one colony that grew negative gram stains. But there also were about 2 or 3 that were just below the threshold. So it was detected, but just below the threshold. And this would be a prime example of where you have that flexibility to look further into the data to determine how you might potentially modify that threshold. And then this table here, looking at the overall characteristics. So of the 50, as I mentioned, 34 were considered to be infection, 16 were not. The overall clinical sensitivity based off of these findings for the NGS assay was 54.6%, and specificity was 5.7%, which actually correlated quite well with other literature, with other findings in the literature. And the NPV and PPV were in the 70s. And then when I looked at the analytical performance itself, the sensitivity, when you're comparing it just straight to culture, was 79.2%, and specificity was at 100%. And this is just an example illustrating just we sequenced one tissue. It was an explant RVPA conduit that we tested. And you can see just the abundance of the lactobacillus that was detected by NGS compared to the negative control. You can just see a bunch of random. So overall, automated, targeted genomic NGS directly from specimens. The greatest thing about it with regards to the GenNexus system is that lab in a box approach, where from the specimen, everything that is illustrated in this middle picture here is all done within the instrument itself. And it allows for that reduced turnaround time of even about 10 to 2 days, as I estimate, but even up to 24 hours, depending on the timing that it's set up. The AmpliSeq targeted NGS panel, it does offer on the Ion Torrent GenNexus integrated sequencer, it does offer quite a promising approach to to pathogen detection and to identification. So in summary, NGS has the potential to be an integral part of the clinical laboratory for pathogen detection. To maximize the impact of overall NGS, though, there really is the need to shorten that turnaround time. And the automation of NGS assays will potentially allow for that broader adoption in the clinical setting, so that it's not reserved for the larger labs that currently have the infrastructure to perform these high complexity testing. And with that, I'd like to just list off some acknowledgements here that have really helped us this far with the study, particularly my lab staff, but also the staff in the clinical microbiology. A shout out to Dr. Lucas Osborne, who was the clinical fellow that worked on this study and really led and made the study happen. And then from the informatics side, Dr. Hakim Javadi, who really worked and developed this great bioinformatic tools that we can use to pair it with the GenNexus system. So with that, I thank you for listening and would be happy to answer any questions later.
Thank you, Dr. Jennifer Dien Bard. for your informative presentation. We will now start the Q&A portion of our webinar. So to our audience, if you have any questions you'd like to ask, please do so now. Just type them into the Ask a Question box and click Submit, and we will answer as many of your questions as we have time for. So Dr. Dien Bard, we have quite a few questions that have already come in, so I'm just going to dive in here. Let's see. Let's start with this one. Hello, ma'am. How will you differentiate between malaria and babesiosis disease, as the disease-causing microbe has similar structures?
So our current assay that we pursued only targets bacteria and fungal targets. So it does not include targets for parasites, including Babesia. However, working with the Thermo Fisher team, there are opportunities to kind of explore and expand those panels, and have it designed so that there are custom panels that may target specific parasites, including Babesia.
Very good. Thank you. Let's see. Next question. Let's see. Let's do this one. What is your opinion of comparing the clinical utility of NGS versus the syndromic testing?
So it's a really good question, and I am a huge fan of both, and I think that there is specifically a role for syndromic testing as alongside of NGS. Currently, when you're looking at sterile sites, aside from your CS staff, blood, there's BALs, and I think there's also joint fluids. There's still a need for syndromic testing in other specimen types. But for the ones that are available, I do see it as very complementary to NGS, because with syndromic testing, it is very fast. You can get a result out within an hour or so. So it would be really interesting just to even pursue a study to determine the impact of syndromic paired with NGS, compared to just standard of care method, and also to compare what syndromic testing, because it is more targeted, would detect compared to NGS, and what NGS would supplement as well.
Thank you. Now, our next question is two parts. You mentioned Thermo Fisher Genexus being a game changer. How is it able to provide faster 16S analytics than before? And what is special about the Thermo Fisher 16S analytics compared to, say, Illumina?
I do want to clarify that when I refer to it as a game changer, I was referring to the automation that incorporates the wet lab portions, including the library preps. With regards to the analytics, I'm by no means an expert in that area and cannot really speak to that. I'm sure there are specific differences or similarities, but really what I was referring to for game changer was the actual the wet lab hands-on portion prior to placing it into the instruments.
Next question. Thanks for sharing. Did you use your RefLab NGS sequencing to compare your results?
For the purpose of the study that we published, we did use our reference lab for just a small subset of samples that were discordant. So using that as kind of the orthogonal, I guess just discordant analysis. And what we found was that I believe there were 5 or so that we sent out, and 4 of which were actually corroborated with the NGS findings on the Genexus system. The one that did not was a very low positive. Even on culture, there was, I believe, only a couple colonies or so that grew, and the gram stain was negative. So overall, it did corroborate quite well. But also for the interest of costs and stuff like that, we did limit just to the discordant samples that was available.
Thank you. And following up on that question, how did you validate this testing for clinical use?
So the 50 specimens that were included in the study that we published were also included in our validation. We, of course, also pursued additional testing, including determining limit of detection, precision studies, etc. Basically all the parameters that would be required to validate a lab-developed assay within the clinical lab.
Thank you for that excellent presentation. How many samples could you analyze?
So because we are a relatively smaller lab, we're not a reference lab. We and the send out volume that we have currently for this type of test is lower. It's not in the thousands a day or hundreds a day. So the nice thing for us, and one thing that we really liked about the GenX's system, is that we could test very small numbers of samples, and we calculated that with minimal waste, we can test up to a minimum of 4 samples at a time and a max of 16 samples at a time. That incorporates the total automation portion. And I'm particularly even interested in the 4 samples at a time that we can test, because that would allow us to even do daily, every couple of day testing to improve that turnaround time with minimal waste in reagents and, of course, increase in costs.
Thank you. Our next question, can you also get antimicrobial drug resistance information in this assay?
So for this specific assay, because we're only targeting, 16S and some of the other targets for bacterial or fungal and mycobacterial identification, it does not detect resistance markers. However, we are currently working on pursuing whole genome sequencing on the system itself, and with that, it would be capable of detecting resistance markers and other key just findings as well, including strain and outbreak investigations, for example. But for the purpose of this test, because it really is to identify the pathogen itself, we did not pursue that as an option.
Now, do you think the amplified method could be used to test responses to antimicrobial therapies after starting a treatment?
So this is a very interesting question. I would say that the test that we have in place, no, because we are again just detecting the pathogen itself. And typically speaking, we usually do not encourage performing repeat testing of these as a test of cure as well. We would, because of the fact that you're detecting genetic material, you would expect that it would be present for a prolonged period of time, even when the organism has been eradicated by antimicrobials. So this wouldn't necessarily be the purpose of this test. However, if you're looking at other assays that could potentially explore responses to treatment or so, I would say, I would recommend reaching out to your Thermo Fisher rep and seeing what options there are, and kind of working with the team to explore your needs and what they can potentially offer or help you with.
Thank you. Now, did you detect any bacterial or fungal species without any available treatment options? And what would you do if this happened in a clinical setting?
So all the bacteria and fungus that we did detect, fortunately enough, did have available treatment options. I guess I'm going to interpret this question to determine whether the patient was on the appropriate therapy. And I think that's really the purpose of this test, is if we can get the result out in a timely manner so that it does allow for even escalation of the patient to a more appropriate antibiotic or antifungal agent. And then also on the other hand, to be able to de-escalate them to a narrower drug, just to avoid the unnecessary treatment of a broad spectrum antimicrobial agent. But for the subset that we studied, I believe they were all on appropriate therapy already.
Can you describe how hard it was to get up and running with a custom assay on the GenX's?
So I would say, with full disclosure, that we are a clinical microbiology lab that has performed and offered
many high complexity PCR tests, including viral testing, bacterial testing, but all mainly PCR. And it really was just during the COVID-19 pandemic that we were able to explore next generation sequencing in our lab using the GenNexus system for SARS-CoV-2. So we had some experience, but I am not an expert even when it comes to the post-analytical processes. So I would say that I had really great individuals that worked in our lab. Our clinical fellow that was amazing, the bioinformatician that I mentioned to you, but also just the overall the Thermo Fisher team and just the support. Shout out to Eric from Thermo Fisher, who was and still is extremely supportive and helpful in helping us troubleshoot and investigate any outliers or anything like that. So overall with that, the experience was very positive, although it was a huge learning curve for us. And we're not shying away from it because, as I mentioned, we are exploring a whole genome sequencing assay as well right now.
Now, do you think your NGS test will become adopted as a standard method in clinical microbiology labs in the future?
I would hope so. And, I think this was, I guess, if you relay it back to even just PCR testing, which in, a couple of decades ago were mainly in labs that had the proper infrastructure to perform high complexity PCR tests that required multiple steps, manual extractions, manual PCR setup, etc. And although this still is the case in some labs, I mean, the availability of these sample to answer PCR technologies, that offer syndromic testing, things like that have really simplified PCR, made it moderately complex instead of high complex, and offered and allowed it to be incorporated into many clinical labs that wouldn't have thought that they would ever be able to perform PCRs in their lab and still wouldn't be able to perform high complexity testing. So in the way, this is how I kind of see this approach for NGS, to automate it to the point where you can have it as a moderate complex type of test, where it can be offered in the routine clinical microbiology labs where it really ultimately belongs.
Thank you. And following up on a previous question, what are the footprints of the machine used as POC?
The footprint of the machine, so it's actually the one that we have is it's not a bench top. So it's basically from the ground up. And, I apologize, I don't know the exact measurement, but it is a relatively tall, probably my height, 5'2" or so, big machine. Can it be used at the point of care? I would say no, and I would really caution against that, at least at this point. It still requires, an extraction to happen outside of the instrument, which needs to happen in a proper infrastructure lab that can do the extraction. Perhaps one day when they can all be incorporated into one, but at this point, it is not ready for point of care at the clinic level.
Thank you so much. And we have time for one last question. Finally, what is the best way for someone in our audience to contact you if they have questions about this assay, getting it up and running in their lab?
Well, I am very happy to answer any questions that others may have that may come up. I'm available through LinkedIn. I'm very Googleable, I guess. If you just search my name, you'll see. You'll even find my email that way as well. But LinkedIn works.
Excellent. Thank you so much, Dr. Jennifer Dien Bard, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for sponsoring today's educational webcast. Before we go, I do want to thank our audience for joining us today and their interesting questions. We had quite a few questions we were unable to answer, and additionally, those that come in during the on-demand period, we will get those answered via the contact information you provided at the time of registration. Today's webcast can be viewed on demand, and LabRoots will send you an email that will alert you when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Dr. Dien Bard, thank you so much again for joining us. Till next time, everyone, have a great day. Bye-bye.
Thank you.